Hello students, let's go ahead. Objectives are LD50 and factors that influence the effectiveness of chemotherapeutic agents. Let's see LD50. What is LD? It is lethal dose. In toxicology, lethal dose is an indication of the lethal toxicity of a given substance or a type of radiation. Okay? Now, lethal dose is represented or recorded as dose per kilogram of subject body weight, at which a given percentage of subjects will die. Understood? Now LD50 is also called as median lethal dose. Okay? Lethal dose 50%. Next. Now, the test was created by J.W. Trivan in 1927. So, the term semi-lethal dose is occasionally used with the same meaning. Okay? So, it can also referred as sublethal dose. Now, because of this ambiguity, it is usually avoided. So, what is used? LD50. It is usually determined by tests on animals such as laboratory mice, rats. Fine. In 2011, the US Food and Drug Administration approved alternative methods to LD50 for testing the cosmetic drug Botox without animal tests. Fine. Now LD values for humans are best estimated by extrapolating results from human cell culture. Okay, practically they were best used. Next. Now one form of measuring LD lethal dose is to use animals like mice or rats, as I told you, converting to dosage per kilogram of biomass and extrapolating to human norms. Now what happens? The degree of error from animal extrapolated LD values is very large. The biology of test animals differs in important aspects to that of humans. So, for instance, mouse tissue is approximately 50 times less responsive than human tissue. Okay? Understanding? Next. As I told you, LD50 is usually expressed as the mass of a substance administered per unit mass of test subject, typically as milligrams of substance per kilogram, but stated as nanograms, micrograms, milligrams or grams. Okay, depending on different subjects. Now stating it this way allows the relative toxicity of different substances to be compared and normalized for the variation in the size of animals exposed. Although toxicity does not always scale simply with body mass. Clear? Now lethal dosage often varies depending on the method of administration. For instance, many substances are less toxic when administered orally than when intravenously. Understood? Now for this reason, LD50 figures are often qualified with the mode of administration. For example, LD50 IV. Okay? So it is given as like orally administered or intravenously administered. Fine. This was LD50. Next, factors that influence the effectiveness of chemotherapeutic agents. Now, treatment of a diseased bodies by administering therapeutic drugs or antimicrobial agents is not that simple. Now, we also know that there are variety of ways by which drugs are administered in the body. And also we know that the drugs do not always spread that rapidly throughout the body or immediately kill all the invading pathogens. Okay, so during all these practices, a complex factor influence was like array was seen. What was that? The main factor, mode of administration of drug, and second one was pathogen susceptibility to drug. So let's see mode of administration of drug. The drug must actually be able to reach the site of infection. The mode of administration plays an important role. For example, a drug such as penicillin G is not suitable for oral administration because it is relatively unstable in stomach acid. Some antibiotics, for example, gentamicin and other aminoglycosides, are not well absorbed from the intestinal tract and must be injected intramuscularly or given intravenously through veins or through muscles. Understood? Now some more examples. Antibiotics like neomycin, bacitracin, they are applied topically to skin lesions. And non-oral routes of administration often are called parenteral routes. Even when an agent is administered properly, it may be excluded from the site of infection. Now let's understand with an example. Body clots or necrotic tissue can protect bacteria from a drug. How? Either because body fluids containing the agent may not easily reach the pathogens, or because the agent is absorbed by material surrounding to it. Understand what we are talking on? Okay. Next, pathogens susceptibility to drug. The pathogen, it must be susceptible to the drug. Bacteria in abscesses you understand now. Bacteria in abscesses may be dormant and therefore resistant to chemotherapy. Now see example, because penicillin and many other agents affect pathogens only if they are actively growing and dividing. Understand? Okay. A pathogen, even though growing, may simply not be susceptible to a particular agent. For example, penicillins or cephalosporins, which inhibit cell wall synthesis, do not harm mycoplasmas, which lack cell walls. Understood? Pathogen susceptibility to drug. And the third one is pathogens MIC value. In the last lecture, we saw what is MIC. So let's see. The therapeutic drug, it must exceed the pathogen's MIC value for its greater effectiveness. Okay? The concentration which is reached will depend on the amount of drug administered or the route of administration and the speed of uptake and the rate at which the drug is cleared or eliminated from the body. Understood? It makes sense that a drug will remain at high concentrations longer if it is absorbed over an extended period and excreted slowly. Understood? Now let's see one more point, bioavailability of drugs. A substance will only take effect if it can be absorbed by the body. So, bioavailability is the key to creating a supplement that delivers proven benefits. Understand? It's a measure of how easily a substance can be absorbed by the body. Okay? Next, bioavailability of a drug is largely determined by the properties of dosage form, which depend on its design and manufacture. Differences in bioavailability among formulations of a given drug can have clinical significance. Thus, knowing whether the drug formulations are equivalent is essential or not. Understood? Now let's see causes of low bioavailability. Orally administered drugs must pass through the intestinal wall and then the portal circulation to the liver. Now both are common sites of first pass metabolism, means metabolism that occurs before a drug reaches systemic circulation. Thus, many drugs may be metabolized before adequate plasma concentrations are reached. Low bioavailability is most common with oral dosage forms of poorly water soluble, slowly absorbed drugs. Fine. Now there are many criteria like age, sex, physical activity, genetic phenotype, stress, disorders, if there are any disorders, or previous gastrointestinal infections. Okay, surgeries can also affect drug bioavailability. Chemical reactions, they also reduce absorption and can decrease bioavailability. They include formation of a complex, for example, between tetracycline and polyvalent metal ions. Hydrolysis by gastric acid or digestive enzymes, example penicillin and chloramphenicol, palmitate hydrolysis. Understanding these reactions or the complex. Next, here you can see plasma drug concentration upon time. What happens? Plasma drug concentration increases with extent of absorption. The maximum plasma concentration is reached when drug elimination rate equals absorption rate. Bioavailability determines based on the peak plasma concentration can be misleading because drug elimination begins as soon as the drug enters the bloodstream. Understood? Now peak time means what over here? When maximum plasma drug concentration occurs is the most widely used index of absorption rate. The slower the absorption, the later the peak time. Now you get it? For drugs excreted primarily unchanged in urine, bioavailability can be estimated by measuring the total amount of drug excreted after a single dose. Ideally, urine is collected over a period of 7 to 10 elimination half-lives, you can say, for complete urinary recovery of the absorbed drug. You're getting this is an example. After multiple dosing, bioavailability may be estimated by measuring unchanged drug recovered from urine over a 24-hour period under steady state conditions. Understood? Understood bioavailability? Okay. If you have any doubts, do ask me. Thank you.